Okay, it's always a hard act to follow Melissa. So thank you, organizers, for putting me up against her. But I'm really pleased that she talks so heavily about macrophages and Anna. I think we should call this macrophage revolution by the time I think we're done with all of the speakers today. And thanks, Edmund, for the introduction as well. So I'm very excited to be here a year after we kicked off, and we were so happy to finally get this grant and to report on some progress and share the perspectives and work that we've done in osteoarthritis. And I, too, will be presenting a macrophage perspective. So would it surprise you to know that there's more Canadians with arthritis versus diabetes, stroke, heart disease, cancer, and dementia put together? It's a silent disease, and this number is growing, right? And it's a huge burden for our society as our population ages and also gets more obese. And unfortunately, we have no treatments for this. Patients and this disease is simply managed, and much of the management is really pain management, and a lot of it is opioid prescription. So that has really fueled the opioid crisis. And this is not because of a lack of effort or investment in this field. Many of these singular therapies that address one aspect of this very complex disease have failed to reach endpoints in clinical trials. And this is because our understanding of this disease has been fundamentally incorrect. We've thought of this for a long time as an old person, wear and tear, mechanical cartilage disease. We now understand that this is a whole organ, multi-joint tissue disease that involves the muscle, the tendon, the ligament, the fat, the bone, the cartilage, yes, and also the synovium, which contains, yes, macrophages and fibroblasts. And we also now know that osteoarthritis is really a metabolic disease. Obesity is the #1 risk factor for getting osteoarthritis. And we also know that this is a low-grade chronic inflammatory disease with all the stresses that chronic inflammation brings. So I want to share with you this synovium membrane, which is this lining of the joint, and it's not any longer considered a passive bystander, but actually an active driver of pathogenesis in OA. It's a nutrient supply to the entire joint because it's the only part that's actually vascularized. And during OA, this synovium membrane actually thickens, and there is more blood vessel supply, which brings in more infiltrating immune cells to the joint, and that causes more pain and inflammation. When you look at the synovium, there's a lining, and then there's a sublining layer, and there's many different cell types, but key among these are fibroblasts and, yes, macrophages. When you sample the synovial fluid, this is the fluid that bathes the entire joint, and it's actually, again, the lubrication for the joint, but also the nutrient supply. This also contains many immune cells, and yes, you'll find B cells and neutrophils in an arthritic joint, but also, again, monocyte and macrophages are by far the most prevalent immune cell in the joint. Elegant studies that have come out of Virginia Krause's lab has used radioisotopes to label folate receptors on macrophages in knee OA patients. And what she has shown is that the presence of these activated macrophages that express these folate receptors correlates with more severity of OA and more pain in these patients. We've also shown that monocytes and macrophages in both the synovium and in the synovial fluid come in a spectrum of flavors, but this intermediate CD14, CD16 subset, when this increases in frequency, patients feel worse. There's more pain, there's poor quality of life, and there's more stiffness in their joints. These macrophages, we think, are key drivers of the synovitis cascade, which is really underlying the pathogenesis in OA. So these macrophages get activated, and by the way, they get activated by many things, including the gut microbiome. So there is this huge cross-section between the gut microbiome and the joint, which my lab is exploring, which I won't get into today, but that causes a lot of metabolic dysregulation in the macrophages, and that causes them to be functionally perturbed. And one of their key roles in the joint is really to vacuum up the debris in the joint. So the joint is very much an active remodeling environment. New cartilage and collagen extracellular matrix and proteoglycan matrix is made, but it's also degraded. And this is in nice equilibrium during homeostasis, but this equilibrium shifts obviously to more catabolism during osteoarthritis. But it's further exacerbated because all of these fragments, these ECM fragments that are generated, begin to accumulate because the macrophages are also impaired in their functionality and are not able to clean up this debris. So this accumulating debris then activates more proteolytic enzymes, which drives more cartilage degradation, which creates more DAMPs, PAMPs, alarmins, reactive oxygen species in the environment, which then pulls in more infiltrating immune cells, and that drives this very vicious self-amplifying cascade of synovitis. And I think underlying all of this is the mitochondrial dysfunction within the macrophages. And we know that they're dysfunctional because they're releasing mitochondrial DNA, they're releasing ROS, we know that their lipid metabolism pathways are shifted, their glycolysis, they're using glycolysis for their respiration. So the whole energy profile of these macrophages is shifted. Now, mitochondrial dysfunction has been studied in the context, I would say, of chondrocytes and autophagy, but very little has been looked at in terms of macrophages and fibroblasts in the synovium, and that's really the key focus of our project within the grant. So one of the first questions we asked is, okay, what does the OA environment do to monocytes and macrophages? And so to answer this, we took healthy monocytes from individuals, or we took synovium macrophages from end-stage patients that were getting a transplant, and we exposed them to synovial fluid, and we did a series of readouts. I'm only going to show two in the interest of time. One was phagocytosis, so we looked at their ability to phagocytose, and we showed really nice impairment, maybe not, impairment, impairment with the synovial fluid that was added to the second and the fourth, I'll let you look at it. The second and the fourth one, you can see that there is a drop in the phagocytic capacity of both healthy monocytes, but also of already diseased macrophages when they're exposed to the synovial fluid environment. When we look at mito-ROS production, we again see healthy monocytes, but put in a synovial fluid environment, we have an increase in their mito-ROS. The synovial macrophages, there isn't that much of a shift, but I want to point out that these are very different baselines. I've just normalized it to put it on one graph, but they start off in a very different place. But this was very interesting to us because it said that there's something about the environment that is causing this functional and mitochondrial disruption to these monocytes and macrophages. So Raksha Devi, postdoc in my lab, is leading the work. So she's going to be looking at, well, okay, can we transplant healthy mitochondria into these macrophages and rescue both their mitochondrial as well as their functional impairment? And in turn, will that then change their energy and functional profile and their crosstalk with all of these other tissues within the joint and mitigate this cascade that I talked to you about? Now, we can source mitochondria from different cell types, and we're going to be hearing a lot more about this later today, especially from platelets, but my lab works with mesenchymal stromal cells, so that's where we sourced our mitochondria from. So in preliminary proof of concept experiments, Raksha took healthy mitochondria from MSCs and added them to CD14 macrophages, and she saw a reduction in mito-ROS, this is in blue on one axis, and an improvement in their phagocytic capacity, which is shown in red in the other axis. She also repeated the same experiment but used a synovial fluid matrix. This is a non-OA synovial fluid matrix that we can obtain from cadaveric donors. And again, she sees, even in this matrix, that there's a reduction in mito-ROS in blue and an increase in their phagocytic capacity in red. Now, this is just proof of concept data. A lot more optimization needs to be done in terms of dosage, duration, and source of the mitochondria, so this is preliminary data. And we also need to move to more of a diseased tissue, so we need to move to our synovium macrophages with the synovial fluid, so we are working on that. But I want to take a little bit of a detour and highlight some of the story, and it's very interesting to me that Melissa just talked about primitive macrophages in the context of cardiac tissue. Here I am talking about primitive macrophages, but in the context of the joint, and again, emphasizing what she said, which is that these are really important cell types. And we know that there are embryonic-derived macrophages that are present during embryonic development. They express a chemokine receptor called CX3CR1, and we know that they persist into adulthood. This is in mouse joints, and they form this almost endothelial-like barrier function, which has a very protective role. During pathogenesis, we hypothesize that this barrier gets disrupted and causes, and is part of what is happening in the OA pathogenesis. So to really chase this, we've been using transgenic mice, which express CX3CR1 and a permanent TAM-inducible TD reporter. So it labels all the CX3CR1 cells and their progeny. And we can surgically induce osteoarthritis in our mice and follow them longitudinally. This is work that's being led by Dr. Shahrzad Nouri in my lab. And what Shahrzad showed is when you look at the three axes, you see a massive influx in infiltrating monocytes. Okay, this was known. But what was very interesting is that many of these monocytes now take up residence and live in the niche as recruited macrophages. The third axis, or the last panel there, shows resident macrophages. These are the embryonically derived CX3CR1 positive, tomato reporter positive macrophages. And their numbers, these are absolute numbers, are relatively unchanged during OA progression. But on a frequency basis, they're essentially getting outdiluted and outcompeted by these recruited macrophages, which are essentially taking over in the joint. Shahrzad's also been very interested in doing site sequencing. So we've been using high-dimensional flow cytometry on these different subsets and then overlaying it with single-cell transcriptomic data. And what she's seeing is that not only are these embryonic-derived macrophages diminishing in frequency, but they're also activating a lot of metabolic and mitochondrial stress pathways, and many of the mitochondrial genes and pathways are being shut down. So this probably explains why this macrophage population is no longer able to exercise its protective functionality during OA. And this is a population that we're very interested in studying, but it's a very rare population that is also diminishing as OA progresses. So like Melissa, we've been using Gordon Keller's embryonic-derived macrophages, which recapitulates yolk sac hematopoiesis. And I won't get into this too much, but just to show you that it also expresses many of these regulatory transcripts, which we have seen in our primitive embryonic-derived joint macrophages. And when we perturb it with synovial fluid, again, we see both a reduction in mito-ROS and an increase in mito-ROS and a reduction in their phagocytic capacity. So this is a great tool for us to now go and study mitochondrial transplant and to understand and tease out some of the mechanisms of what is happening to that primitive joint embryonic-derived macrophages in the joint with this particular cell population. Okay, back to the regularly scheduled program, which is the mitochondrial transplant. And we know, as I said, that crosstalk between multiple joint tissues is really important. And we have a well-established joint-on-a-dish model system where we take end-stage patient cartilage and synovium, and we can co-culture them and do multivariate gene, protein, and functional readouts simultaneously. And so the system is well established and validated, and to this we can add different doses, different sources of mitochondria, different formulations, and really optimize our transplant process and simultaneously gauge which cell type, so the cartilage or the synovium or the fibroblasts or the macrophages is really uptaking these mitochondria and kind of the efficiencies and the net result of this mitochondrial uptake. And we know this is important, especially with fibroblasts, because they're in close proximity to the macrophages. And preliminary experiments indicate that, yes, when we add mitochondria, they are being uptaken by these fibroblasts, and we see some changes, examples in their ATP and cytochrome B, some reductions in their proteoglycan, proteolytic enzymes, and some small changes in their fibrotic genes. Once we have this optimized, we're going to move to an organ-on-chip system, which we're developing with Edmund's lab. And this is, again, patient-derived. And we have two models, a cartilage-on-chip and a synovium-on-chip. And the cartilage-on-chip is really cool because we can now incorporate mechanical compression, which mimics mechanical abnormal loading, for example, in patients that get a meniscal tear or a ligament tear and then go on because of that mechanical instability to develop OA. So this mimics a mechanical phenotype. And down the road, we'll be adding cartilage strain to this. And we also have a synovium chip model, which incorporates a vascularized network through which we can flow immune cells, particularly monocytes. So we can mimic that infiltration of monocytes and how it affects the various interactions with other cell types. And to this, of course, we can add the mitochondria, and again, we can do multivariate readouts. This is my last slide, just showing you that the model is in place, and Lauren Bond, PhD student co-supervised by Edmund and I, along with student in Edmund's lab has taken this cartilage on chip and applied mechanical compression, and then applied a first-line treatment, an anti-inflammatory treatment, which is a corticosteroid that's often given to OA patients, and shown very nice rescue in mechanosensitive, catabolic, inflammatory, hypertrophic genes. And what she's also been able to show is that this happens very much in a patient-dependent, donor-dependent manner. So if you take 4 different donors and you look at their proteoglycan content, under inflammatory or mechanical stress, you see a very different proteoglycan profile. And when you give them this corticosteroid treatment, you see a very different response. So this mimics some of the heterogeneity that we have from one patient to another within the model. So the model system is ready. And so we are, I think, hopefully I've shown you, along with Melissa, that macrophages are really important in knee OA. Not only do they drive the pathogenesis, but they in turn are also perturbed by the environment and also suffer the dysfunction both on the metabolic and mitochondrial level. And preliminary proof of concept experiments suggest that yes, they are uptaking the mitochondria, and there is differences both in their mitochondrial and functional readouts, and we can now take these to our more complex in vitro, static as well as dynamic models, and before we move to our in vivo studies. I would like to thank the collaborators and of course members of the lab and the New Frontiers for funding this work. Thank you.